I'd like to thank Dr. Tam and Dr. Anders for inviting me. And as was pointed out, I'm going to focus on supportive cancer care and specifically mucositis. But I'd like to start the talk by talking about some of the new dosimetry principles that are really helping us do more precise photobiomodulation dosing, and then move into the actual evidence for the others. These are my disclosures. I consult for a couple of these companies, and I hold office in some of the organizations you will notice that are dedicated to bringing photobiomodulation into mainstream medicine. So let me begin by talking about dosing. I think this audience does not require an introduction to dosing, but most of our photobiological dosing principles are linear in nature, and these have been well characterized, whether you're looking for a deeper pigment tanning in this particular case, or you're trying to do very simple linear dosimetry for many of the photobiological responses. They are premised on the fact that you have effective absorption, effective energy transfer, and a certain biological change. If you're familiar with photobiomodulation, you have heard about this very famous curve called the Arndt-Schulz curve. And we make the analogy that in photobiomodulation dosing, unlike a simple linear change, we are focused on something that is akin to something that we can do with a common protein, right, albumin. And if you use the right amount of dose, both in terms of irradiance and treatment time, we can actually make many different delicious dishes. And the simple way to think about it, and I do a lot of pre-doctoral teaching, is that we talk about how we can make all these dishes if you can unfold the protein in a particular way, right? And that's what you're seeing in this particular dishes. So the non-linearity has led our field to begin focusing beyond absorption. So we know that absorption is really important for energy transfer, but now we have started paying more attention to inelastic scattering. And it's turning out to be a huge game changer for reasons that I like to elucidate based on that concept. So the way we think about PBM dosing now is how are we transferring energy into the system? And this is the first step. And the second step involves how are we transforming that energy into a biological effector. The energy has to do something within the biological system for it to be effective. And we know that heat is not a part of photobiomodulation. Thermal changes, and we have a particular range above 45 degrees, and I'll show you the data, it does not contribute to the photobiomodulatory therapeutic effects. Largely, it is biochemical, photobiochemical, as well as photophysical, biophysical changes that are contributing to the therapeutic benefits. So when we start thinking about photobiomodulation dosing, we know all of us have used different wavelengths of light. It is amazing that there are so many wavelengths of light that are capable of photobiomodulation, and that usually gets us into a pickle with the physicists and other groups. But we do understand that if we start studying photon distribution in three-dimensional volumes, especially heterogeneous biological system, we have an expert in the audience here, it gets very complicated very quickly, right? So to go away from that concept and improve our consistency in clinical dosing, we have started focusing on what is the energy on the surface. And again, this audience is very familiar and very used to thinking in those terms, so this is not a new concept for you guys. But in photobiomodulation, that has been a huge sea change, because that is something that a clinician can pay very careful attention to, the tissue surface irradiance. And that's largely because of the simplicity of being able to measure this consistently and report it. So when we talk about the evidence for supportive cancer care, there are so many good studies, but they still don't report the dose accurately. So this concept of just reporting the surface irradiance has been a huge step forward for our communication of PBM dosing, which I like to emphasize. So as you know, we can think about fluence in terms of irradiance and time. The additional aspect that we have started paying a lot of attention to is the wavelength photon energy. And we think of this as a minor, if all of you, I'm sure, are familiar with the amount of minuscule energy that each photon carries. But when you start thinking about photobiomodulation, which is a low dose phenomena, that energy becomes really important. And if you're familiar with PBM dosing, simply adding that photon energy now has enabled us to do more precise calculations and more precise clinical outcomes. So the concept of photon fluence is simply adding the photon energy to your classical irradiance and time calculations. The third step we very quickly realized turns out to be how do we compare different devices which have different wavelengths, and especially people who are combining different wavelengths to do different kinds of treatment. And that is where, as a field, we have agreed that we will use 810 nanometer as a reference wavelength. And by doing that, we simply divide our photon fluence by 4.5, and we use this term Einstein, which obviously we did not invent. Very catchy term, and it has been used for a long time in greenhouses, where they look at efficiency of photosynthesis units, light units. So we actually borrowed this term from that field, and we've been using this in photobiomodulation with lots of good results. So we can actually communicate our dose much more effectively. And the big advantage of doing this, if you can see the cheat sheet at the bottom, is that irrespective of where you are in the world and with which unit that you're trying to do treatment, there is an interconversion factor that is showing lots of promise. Has to be evaluated further, but at the moment, it is helping us communicate our dose in a very effective way. So the clinical implications of the new dose is that we are moving towards an harmonization of dosing recommendations. We are even extending the spectrum of PBM wavelengths. We classically call it 400 to 1200, and now there is evidence, as many of you know, beyond those spectrums. There is also the possibility, if you're using multiple wavelengths, to simply add those dosing calculations and be more accurate with your dosing reporting, as well as your consistency of clinical outcomes. And something that I think we are realizing now, some of this work is still unpublished, is that the transit zone, when you're going through multiple layers of tissue, we were talking in the morning about using different wavelengths, that transit zone where you're transversed by both the photon energies, we have never paid much attention to that. And it's turning out to be really important because the biological outcome of that elevated dose can inhibit or neutralize some of your PBM benefits inside. So that is now something that we can actually plug into many of our calculations. People who do transcranial, you're worried about what's happening when you're going through the scalp and the cranium. Now we are paying attention to that even more precisely. So that's getting to be better. So all of this progress in the field has been tangibly recognized, as we now have clinical practice guidelines in supportive cancer care from the Multinational Association of Supportive Care in Cancer, the International Society of Oral Oncology, and the World Association of Photobiomodulation, WALT. As you're already, I'm sure, familiar, the FDA has recognized photobiomodulation as a discrete category of a form of light treatment, and the guidance document came out last year in January. More importantly, at least as a dentist, the American Dental Association has actually recognized this treatment as a discrete form of light treatment, non-surgical, non-invasive, non-pharmacological. And that's been a huge plus for, as you know, many of our traditional dosing and clinical deliveries based on insurance and recognition by the CMS. And this changes has been a huge milestone for our field. So switching gears a bit, all of this great news in the field of photobiomodulation was largely catalyzed by the evidence that we were seeing with cancer treatment-associated mucositis. So if you're familiar with oral mucositis, it is a condition where you get redness, breakdown, wounds in patients who are getting either chemotherapy or radiation therapy or transplants, graft versus host disease. And based on a systematic review and meta-analysis of 35 multi-center, placebo-controlled, randomized human clinical trials, the MASC, ISO, and WALT have put forth clinical practice guidelines recommending the use of photobiomodulation, both for prevention as well as treatment of oral mucositis. Your cancer patients can get this treatment before they start chemo or radiation. And this is based on, I'll show you some of the data, on the fact that we can increase the molecular resilience in the tissues that are going to be insulted or traumatized with the eventual cancer treatment. So treatment aspect, again, if you're familiar with photobiomodulation, we have known for the largest time that photobiomodulation can stimulate wound healing. And if you have heard about photobiomodulation, the other thing you always hear about is the breadth of its clinical application, right? Traumatic brain injury, Parkinson's disease, Alzheimer's, mucositis, arthritis, and it sounds too good to be true, right? And we have always had a lot of skepticism and cynicism about how can this be. If you think about these diseases, what's common in all of the pathophysiology is the fact that you have pain, you have inflammation, you have an aberrant immune response, and a lack of healing. Photobiomodulation seems to address these fundamental biological responses. And we now understand many of the mechanisms of how this occurs. Most people are familiar with the cytochrome C oxidase mechanism, where this enzyme in the electron transport chain transiently increases ATP and ROS that induces a therapeutic gene expression. We also now understand that there are certain neuronal transporters and receptors which can be transiently and reversibly inactivated to bring about pain relief. TRPV1 stands out among them. And the third mechanism was described by my lab, where we looked at an extracellular growth factor, TGF-beta1, which can be selectively activated with low amounts of photonic illumination. I'd like to elaborate a little bit on that and segue into where we are with mucositis. But we all acknowledge in the field that when you're doing photobiomodulation treatment, you might harness multiple mechanisms in multiple cell types. Light is being exposed to multiple epithelial cells, fibroblasts, macrophages, neutrophils, endothelial cells, right? There's no way of controlling that. So light is activating multiple therapeutic mechanisms. And we acknowledge that that's probably true in many of the diseases that we are able to treat very effectively. So to show you some of the evidence from our lab, we started with some very simple experiments where we are taking, in this particular case, odontoblasts, and we are doing 8-10 nanometer photoillumination at different doses. And as you can see, you can induce reactive oxygen species in a controlled manner, as detected by mitosox red in this particular case. And we are tracking mitochondria with MitoTracker green. So this basically was the first step in our mechanism where we are able to convert photonic physical energy into a biochemical species. We did several other studies. This is one of our key studies where a single amino acid substitution on the latent TGF-beta molecule. This growth factor is secreted. Can you see my arrow on that? Yeah? So this latent complex actually gets activated by dissociating itself from a latency-associated peptide. On the latency-associated peptide, a single amino acid, a methionine, at position 253, when we substituted this oxidizable methionine to alanine, we saw that we were not able to activate this complex. So this is your wild-type complex. I'm sorry, the labels are not showing up. But this is the wild-type complex that is being activated. And when you make a single amino acid substitution, you don't see this activation. So we were very convinced that this is a mechanism worth chasing further. And we actually did several studies. Some of the in vitro studies involved simple scratch assays, where we were plating oral keratinocytes in a confluent layer. And as you know, that leading edge actually moves towards each other and closes. And this is the FDA requirement for good wound healing, right? Intention to close. So if you do photobiomodulation treatment in this particular context with a particular wavelength, you can see that this can be accelerated significantly. And again, we have a poster where we have explored multiple wavelengths that was shown this morning and will also be presented tomorrow by my fellow Deepti. We also know that there are epithelial stem cells in the margins that require to increase in their number. And if you do a simple colony forming unit assay with different doses, we are able to observe increased colonies in number and in the size of these colonies. So both of this suggesting that we are able to increase wound healing if we have treated that with photobiomodulation. So we took this model, and these are control lithomates, where you see the regular latent TGF-beta molecule, which we have shown before that if you do photobiomodulation treatment, this is SMAD2 Western blot, you can see that you can activate this signaling molecule. And surely enough, when you make third-degree burns, full-thickness burns, and do photobiomodulation treatment, you can significantly accelerate their closure and reduce. You can see that these wounds also have less inflammation And macrophages turned out to be very important in this process. This work is published in scientific reports. We did a similar experiment, but we substituted the TGF-beta 1 with another isoform TGF-beta 3. And by making a chimeric transgenic animal model, which turned out to be inactivatable by photobiomodulation treatment. So even though we make the same burn wounds and do PBM treatments, these animals cannot activate TGF-beta. This turned out to be critical for its healing phenotype. So in terms of the burn wound healing, which affects both the immunomodulation via macrophages, as well as the keratinocyte proliferation, migration, as well as increased colony-forming units, TGF-beta seems to be a key mechanism to promote photobiomodulation wound healing. So we have evidence, I believe, at the highest level in biology, and also I showed you some human clinical data that correlate really well. The other question we get often is that, how do you know, because photobiomodulation is such a subtle experience for the patient as well as for the clinician, how do you know you're not doing excessive dosing? And that was this study where we were looking at the role of thermal heat generation in our animal study. So you can see that as you increase the amount of, in this particular case, 810 nanometer laser treatments, when you reach a temperature of 45 degrees, which is not very hot, right? You start seeing beginnings of epithelial damage here in this particular case. And we were very perplexed to see that 45 degrees is causing this damage. Turns out, biologically, at 45 degrees, what you're doing is inactivating ROS scavengers. And here's an example of one of the main ones, catalase. We have done similar experiments with glutathione peroxidase, superoxide dismutase. So at 45 degrees, it's an enzyme, right? And all of us who work in the lab know we can inactivate enzymes at 45 degrees if you warm it. And this is what's happening in the skin. So if you increase the temperature, you may not cause overt damage, but you are beginning to cause redox-mediated damage. And that is why photobiomodulation is a non-thermal event. I get a lot of pushback when I say that because a lot of people say we love hypothermia, right? We love our saunas, but that is a different kind of treatment. That is not photobiomodulation, right? So 45 degrees is where we have maximal PBM therapeutic benefit. And we have done these experiments in animals, and you can see that a single transcription factor, ATF4, in the endoplasmic reticulum drives this phenotype. So if you look at ATF4 levels, it increases in a dose-dependent manner as we deposit increasing 810 nanometer laser energy. But at the highest detrimental dose where we start seeing the damage, you can see that ATF4 expression is downregulated. And this turns out to be really important, and all of this is documented in these two papers, where it actually shuts down the autophagy repair response and pushes it towards a cell death response, which is a combination of autophagy and apoptosis. So at that particular temperature, at the subcellular level, you're actually going to see damage which is irreparable in those cells. However, you can see the trend here, which is showing you the molecular basis for resilience. This is what we think is happening in your cancer patients who are getting better. So you're inducing a low level of stress and preconditioning your tissues at the molecular level, which are then able to withstand your chemo or radiation trauma. So based on this, we have actually started using both the temperature as well as ATF4 when we are able to get biopsies or swabs in patients to look at them as clinical and molecular biomarkers. And all of this is enabling us to do precise photobiomodulation dosimetry in clinical care. The other question we often get is, these are cancer patients, does photobiomodulation increase the tumor burden? And the answer is an emphatic no in all the studies done so far. The first two of them are actually clinical retrospective analysis of patients who are receiving photobiomodulation for oral mucositis, and they have not found increased incidence of recurrences or secondary tumors in these patients. The third study is a tumor study in an animal model where they put oral squamous carcinomas in animals and directly irradiated them with photobiomodulation treatment and did not see an increase in tumor burden. There are also several reports from Wellman and Charleston from your labs actually showing us that there might be also an immune adjuvant effect of light. And that is gaining a lot of attention now because we know that tumor cells have, we have an anti-tumor surveillance mechanism in our host, and photobiomodulation seems to be increasing their ability to fight these metastatic tumors or single clones of tumor cells. So overall, PBM is extremely safe in cancer patients, and there is no evidence that it increases any tumor burden. There are several protocols for photobiomodulation, and again, the jury is out there whether we want to do extraoral versus intraoral photobiomodulation. There are devices for both, or a combination of them, and there are limitations and advantages of each one of those. Most importantly, there are several devices that are already FDA cleared and are available. And there are different formats of devices. So we have dental devices, we have a really nice device for ophthalmology, for dry, age-related macular degeneration, several devices for helmets, or in this particular case, a headset with an intranasal. We have beds, there's one on display here in the exhibits, several dedicated non-ablative laser units, as shown on the top, which are basically non-ablative lasers, and they are dedicated to do photobiomodulation treatment. So if you're interested or if you're new to the field, there are lots of good opportunities right now to actually make a clinical impact in your patients. So I'll conclude by saying we have a very good handle on PBM dosing. We are paying attention to what is our biological target, what is the desired clinical outcome. We are able to precisely prescribe a dose based on the wavelength, the power density, as well as treatment time. And by paying attention to the delivery of the treatment, we are actually able to do persistent, rigorous, reproducible clinical outcomes. With that, I will thank all the people who do all the work in my lab, and I get to present this, funding sources, and several collaborators who have provided all the important transgenic models. And these are some pictures of my lab. Thank you.